I want to start today's session with a bit of a puzzle. It's something you likely see every day, but maybe you've never stopped to ask why it happens.
Okay, I'm listening.
Why is it that you can eat a potato or a slice of bread and your body gets a huge amount of energy from it? But if you tried to eat a piece of wood or grass, nothing happens.
It's a very valid question. And here's the most interesting part. That potato and that piece of wood are made of the exact same ingredient.
That's the part that is so confusing. They're both just chains of glucose, correct?
Yes, identical glucose units.
Why does the body treat them so differently? Why is one dinner and the other just indigestible fiber?
That mystery is exactly what we're going to explore today. We're taking a deep dive into Unit 10, biomolecules. And specifically, we're zooming in on carbohydrates.
A very important chapter for Class 12 students, and I'll be honest, when you open the textbook and see biomolecules, it can feel, well, quite intimidating.
It can. You just see page after page of hexagons and confusing names.
Right. And it feels like there is a lot of memorization involved.
Yep. It often feels that way, but that's the wrong way to look at it. Our mission today is to show you this isn't about memorizing. It's about understanding the architecture of nature.
Architecture.
Yes. If you understand the structure, the literal shape of these molecules, everything else becomes clear. Why sugar is sweet, why wood is tough. It all comes down to the shape.
Okay, I'm ready. Let's start at the very, very beginning. For a student in an exam hall, what is a carbohydrate?
Let's start at the zero level. Forget the complex definitions for a moment. Just look at the word itself. Carbohydrate.
Okay, carbo implies carbon, and hydrate must mean water.
Exactly. Carbon plus water.
Yeah.
Historically, when early chemists first looked at these things, that is essentially all they found. Carbon, hydrogen, and oxygen.
And the ratio was important, wasn't it?
It was. Usually the hydrogen and oxygen were there in the same ratio as water, which is 2.1.
Just like H2O.
Correct.
Yeah.
So the general formula often looks something like COs. To them, it looked like carbon atoms were holding on to water molecules, hence hydrates of carbon.
Okay, that makes sense, but I don't think I can write wet carbon on my exam paper.
Well, you will definitely not get marks for that. That's the old name. To get the real definition, you need to visualize what's happening at the particle level. I want you to close your eyes for a second.
Okay, they're closed.
Imagine a sugar molecule, but don't picture white powder. Picture a chain.
A chain of what?
A chain of carbon atoms, like beads on a string. Now, hanging off each of those carbon beads are other atoms, specifically hydroxyl groups.
The hydroxyl groups, right.
Correct, many of them. And usually at one end, you'll find a specific functional group. It's either an aldehyde or a ketone.
Okay, I remember these. Aldehyde is CHO, and ketone is the C double bond O group.
Precisely. So this brings us to the actual scientific exam-safe definition. Carbohydrates are defined as optically active polyhydroxyaldehydes, or polyhydroxy ketones.
That is a bit of a mouthful. Let's break that down.
It's not as scary as it sounds. Poly just means many.
Many.
Hydroxy refers to the OH groups.
So many OH groups.
And aldehyde or ketone just tells you the functional group family.
Okay, polyhydroxyaldehyde or ketone. But the definition sometimes adds or compounds that produce them on hydrolysis. What does that part mean?
Ah, right. That's because not all carbohydrates are single units. Starch is a huge molecule. But if you break it down with water, that's hydrolysis. It releases those single units, those polyhydroxyaldehydes.
Got it. So it can be the single brick or the whole wall made of those bricks.
Exactly. And this leads to the biggest misunderstanding students have. When I say carbs, what comes to your mind first?
Food, mostly. Bread, pasta, sugar.
Right. Most students think carbs mean sugar. You have to fix that idea. Glucose is a carb, yes. But so is the paper in your notebook. So is the cotton in your shirt.
Wait, cotton is a carbohydrate?
Cotton is almost pure cellulose, and cellulose is a carbohydrate. They're all made of the same atoms.
That's really hard to imagine. My shirt and my sugar are related.
Not just related. They are built from the exact same building blocks. The only difference is how those blocks are connected.
Okay, I definitely need an analogy for this. Because polyhydroxyaldehyde isn't telling me why my shirt isn't sweet.
Fair enough. Let's think about it like Lego blocks.
Okay, Legos. I can work with that.
So imagine a single glucose molecule is one red Lego brick. That single brick is what we call a monosaccharide. Mono means one.
Okay, one brick. Glucose, monosaccharide. Simple.
Now, snap two of them together, what do you get?
Two bricks.
A disaccharide, di for two, like sucrose, which is table sugar.
Makes sense. One brick, two bricks.
Now imagine you have thousands of these red bricks. If If you connect them one after another in a long, long chain, you get a polysaccharide. Poly means many.
A long line of bricks.
And here's the magic. Depending on how you join them, you get completely different things. If you join them in one specific pattern, you get starch. That's your potato. It's edible.
Okay.
But if you take those exact same bricks and join them at a slightly different angle, imagine flipping every second brick upside down before you connect it.
So they would lock together more tightly.
Yes. And you get cellulose.
And that's the shirt.
That's the shirt. That's the wood. That's the plant cell wall.
So it is literally the same material, glucose, but the arrangement changes the property completely. Food versus fiber.
Completely. Starch is for energy storage. Cellulose is for structure.
Why does nature even do that? It seems like it would be confusing.
Actually, it's incredibly efficient. Nature has a lot of carbon, hydrogen, and oxygen, so we figured out a way to use one primary unit glucose to solve two big problems.
Which are?
Problem 1. How do I store energy for later? Answer. Build starch. Problem 2. How do I build a strong wall for my cells? Answer. Build cellulose.
Wow. Using the same ingredients for the food in the kitchen and the walls of the house?
It is the ultimate natural engineering.
Okay, let's get a bit more technical on the joining together part. The exams always ask about the bonds. What is that connection between the Lego bricks called?
That is a glycosidic linkage, or a glycosidic bond. Essentially, an oxygen atom acts as a bridge between two of the sugar units. It's a carbon-oxygen-carbon bridge.
And to make that bond, does something have to be removed?
Yes. It costs one water molecule. To link two glucose molecules, you have to remove a water molecule. That's why we call it dehydration synthesis.
Dehydration for removing water, synthesis for building something.
Perfect. And if you want to break that bond, like when you digest food, you have to put the water back in.
And that's hydrolysis. Hydro for water, olysis for breaking.
You've got it. Dehydration to build, hydrolysis to break. That's a fundamental point.
Okay, so we have Legos, but I want to go back to our starting puzzle, the bread versus the wood. You mentioned flipping the brick upside down. In the book, they say alpha and beta linkages. Is that the same thing?
Yes. And for this, let's use a mental movie, because the pictures in the book are a bit static. Let's imagine a dance floor.
It's the dance floor. Okay.
Imagine it's crowded. Each dancer is a glucose molecule. They want to form a long conga line. That's our polymer.
Okay, a conga line of glucose dancers. I can see that.
For starch, the dancers link hands in a specific way. Let's call it the alpha handshake. It's a comfortable handshake. When they link like this, the line of dancers is flexible. It can curve and coil up like a spring.
A flexible coiling line. That's starch.
Now, for cellulose, the dancers link up using a different beta handshake. To do this, every second dancer has to basically flip upside down.
That sounds really rigid.
It creates a very, very different shape. A tight, rigid, straight line. No coiling at all. And because they're straight, they can pack on top of each other very tightly.
Like stacking logs to build a wall.
Exactly. That's why wood is hard. It's just bundles of these straight beta-link chains.
So starch is a loose, coiling conga line, and cellulose is a rigid, straight security line.
Perfect description. Now, digestion. Think of the enzymes in our stomach as, let's say, gatekeepers.
Okay, and their job is to break the line to get energy out.
Right. Our human enzymes, like amylase, they recognize the alpha handshake. They know exactly how to unlock it. So you eat starch, the enzyme breaks the handshakes, and you get energy.
But what about the beta handshake? The one in cellulose?
Our enzymes look at that rigid, flipped-over connection, and they just don't recognize it. It's the wrong shape. They can't unlock it. So the cellulose line passes right through our body, undigested.
That's brilliant. So the reason I can't eat grass is because my enzymes don't know the secret handshake.
That is it. It all comes down to one key phrase. Structure determines function. A tiny change in the bond angle makes the difference between food and wood.
Structure determines function. I feel like that phrase alone can answer so many reasoning questions.
It's a core principle of all of biology and chemistry.
Okay, so we have the theory, but what about in the lab? For the practicals, how do we see this difference?
Ah, yes. The classic example you absolutely need to know is the iodine test.
Right, the blue-black color test. Drop iodine on a potato slice and it turns dark blue.
Yes, but why? That's the question the examiner will ask you.
I always just assumed it was some sort of chemical reaction.
It's actually a physical thing. Remember the starch conga line? The one that coils up?
The flexible, spring-like one.
Yes. It forms a helix, a coil. Now, iodine molecules are the perfect size and shape to get trapped inside that coil.
Oh, so the iodine is literally sitting inside the starch helix, like a pen inside a spring.
Exactly. It's called an inclusion complex. And when iodine gets trapped in there, the way it interacts with light changes, and it looks deep blue or black to our eyes.
That makes perfect sense. So what about cellulose?
What was the shape of the cellulose line?
Straight, rigid, no coils.
Exactly. No coil means no place for the iodine to get trapped. It can't get in, so no blue color.
See, once you know the shape, the test result is obvious, no memorization needed.
It's a beautiful connection.
And what about technology? Does this have any real-world applications?
It's huge in industry. Think about brewing beer.
Okay, how does that work?
Brewers use starch from barley or corn. But yeast, the organism that makes alcohol, can't eat starch directly. It needs simple sugar, the single Lego bricks.
So they have to break the starch down first.
Precisely. They use a process that activates enzymes, the amylase, to break those alpha linkages. They turn the starch back into glucose. Only then can the yeast ferment it into alcohol.
So the whole industry is basically just industrial-scale hydrolysis.
In a way, yes. It's all about hacking that carbohydrate structure to get to the energy inside.
Okay, let's circle back to the opening question then. Bread versus grass. We know humans can't digest cellulose because we lack the right enzyme for the beta linkage.
Correct.
But cows eat grass, termites eat wood. Do they have some kind of super enzyme?
This is the really counterintuitive part. You might think cows are just biologically better than us, but actually the cow cannot digest cellulose either.
Wait, what? But that's all they eat.
The cow's own body does not make the enzyme cellulose.
Then how are they surviving?
They're outsourcing the work. A cow's stomach is full of special bacteria. It's the bacteria that produce the cellulase enzyme. The bacteria break the beta bonds, and the cow just absorbs the energy.
Wow. So even the cow needs a microscopic helper to crack the beta linkage end.
It just shows how specific these molecules are. Nature has locked that energy behind a specific bond, and you need the exact key to open it.
That's fascinating. Okay, we've covered a lot. I want to switch into exam mode now.
All right, let's do it.
We need the precise language for the board exams. Scenario 1. A question asks to classify carbohydrates based on hydrolysis. What do I write?
You must write about the units produced. First, monosaccharides, single units that cannot be hydrolyzed further. Example, glucose.
Okay.
Second, disaccharides or oligosaccharides. These yield 2 to 10 units on hydrolysis. Example, sucrose. And third, polysaccharides. These yield a large number of units. Example, starch.
Simple enough. Now, what about reducing sugars? This one always confuses students. What is it reducing?
This is a classic question. A reducing sugar is one that has a free aldehyde or ketone group.
Free, meaning it's not locked up in a bond.
Exactly. If that group is free, it can react. It reduces mild oxidizing agents like Fehling's solution.
So if I add Fehling's solution to glucose, I get that reddish precipitate. That's a positive test.
Correct. So glucose is a reducing sugar.
And what about a non-reducing sugar?
Sucrose, table sugar. In sucrose, the functional groups of the two units are linked together in the glycosidic bond. They aren't free to react. So sucrose is a non-reducing sugar.
That's a very common trick question. Why is sucrose non-reducing? The answer is because its functional groups are involved in the bond.
Spot on.
Okay, one more. The structure. We talked about the chain, but what about the cyclic structure, the hexagon in the textbook?
Right. In water, the straight chain actually curls up and bites its own tail. The carbon-1 connects to the carbon-5 through an oxygen. That forms the ring.
And that's where alpha and beta come from.
Yes. When that ring closes, the HOH group at carbon-1 can either stick down or stick up. If it's down, we call it alpha. If it's up, we call it beta.
Alpha down, beta up.
And that tiny little difference, that up or down, determines whether you get starch or cellulose.
It's incredible that a microscopic detail decides if you're eating a potato or a tree.
It really is remarkable.
Okay, let's wrap this up. Quick recap for a student walking into the exam. What are the 3 or 4 absolute must-knows?
First, the definition. Carbohydrates are optically active polyhydroxyaldehydes or ketones. Remember polyhydroxy, many OH groups.
Got it. Second?
The structure-function link. Remember the Legos. Alpha linkages make starch, which is helical, and gives the blue iodine test. Beta linkages make cellulose, which is straight, rigid, and doesn't.
And the third thing?
The bonds. Glycosidic bonds link the units. Dehydration synthesis makes the bond water out. Hydrolysis breaks the bond water in.
Fantastic. I feel so much clearer on this unit now. It's less about memorizing hexagons and more about seeing the logic.
The logic is beautiful once you see it.
Before we sign off, I want to leave our listeners with one final thought. We talked about how humans can't digest cellulose, But imagine,
You know what?
if enzymes are just keys that fit specific locks, and we're getting very good at bioengineering, what if we could make a pill? A pill with the cellulase enzyme that could survive in our stomach.
That is a very provocative thought.
Could we end world hunger? Could humans survive by eating grass or sawdust or old newspapers?
It would certainly change the grocery store. But from a pure energy standpoint, the calories are there. We just don't have the key to unlock them yet.
Something for all of you to think about. You're not just memorizing chemistry, you are learning the source code of life itself.
Well said.
That's it for this deep dive into carbohydrates. Good luck with your studies, keep visualizing those dancers, and we'll see you in the next one.
Keep learning.